Well, welcome everyone to one of the final sessions of the 12th World Stem Cell Summit Public Day. And so far the reviews are in and they've been A+. At least no one wants to tell me if they didn't enjoy it. So I'm very happy as the founder and co-chair of the World Stem Cell Summit. My name is Bernard Siegel. I'm also executive director of the Regenerative Medicine Foundation. So I had the pleasure of meeting Jeff Karp for the first time at a conference in Paris. How about that? That's right. I remember. It's good. So, you know, what a great place to meet anyone. But Jeff really impressed me with the depth and breadth of his knowledge as it relates not only to regenerative medicine, but tissue engineering. And indeed, he is an inspiring figure. You may know him, of course. He's with the Harvard Stem Cell Institute. He's an affiliate professor at MIT. I've read about him. He is a bio-inspired medical problem solver. He is an expert on tissue adhesives, biomedical engineering, and mesenchymal stem cells, among his many talents. But he's speaking to us today about bioinspiration and innovation. And I had the occasion to see a talk online, and as soon as I saw that, I knew that this is something that had to be live streamed, it had to be preserved to the audience of the World Stem Cell Summit around the world. So I'm very happy to present my friend Dr. Jeff Karp.
Thank you. Thank you. Thank you for the nice introduction. So today what I thought I would do is rather than just kind of show data, I really want to talk about the process. And I would say that, in basic science, when we're doing discovery work, often, 9 times out of 10, we conduct an experiment and it fails. And I think the challenge is, how do we take the basic science and advance it towards the clinic? And I think anyone who's ever tried to translate anything, I think, would agree that the number of challenges, the number of problems that one encounters actually grows exponentially. And so we've really tried to make a strong commitment in my laboratory to not just publish papers, but to try to advance that basic science into therapies that can help patients in the shortest period of time. And I would argue that in many ways we're actually biased against solving tough problems. And I don't know how many people here have ever seen, there's this TED Talk by Ken Robinson. It's actually the most watched TED Talk of all time. Have people seen that? There's a few people raising their hands. If you haven't seen it, go watch that talk. It's very entertaining. But there's a very powerful message there, which is what Ken does is he describes that when children are born, every child is born creative. But yet the education system over time actually basically takes that creativity out of kids. And I think that this really negatively impacts our ability to solve problems. And I would argue that what has happened is when we go to solve a problem, we approach it, we encounter a failure, we step back, and then we think we're being creative, and we approach the problem again, but we're approaching it from the exact same angle. And we expect different outcomes, and that never happens. So to really emphasize this point, what I wanted to do is share with you this example, which is a problem. This really happened. This is in Europe. And you see here that we have a car that's fallen into the water. And we have the problem solvers that are lined up trying to figure out, what they're going to do. And they probably have, 2 or 3 different options. And so what they did is they bring in this crane. And things seem to be going well until the following happens. So one problem has become 2 problems. And the second problem is even bigger than the first problem. Something I'm sure we can all relate to perhaps on a daily basis. So the question is, what are the problem solvers going to do next? What do you think? Go home. That's right. They're going to do what most of us, except this gentleman here, would do, which is go and get a bigger crane. And what do you think happens? The same thing. So to me, the challenge is, how do we break free from this repetitive thought process? And I would argue that our brains like to operate in these super low energy states where they anticipate what comes next, right? One example, if this has never happened to you, is this happened to me many times, is that you have your cell phone, it tells you to update your password. So you put in the new password, and then the next time you go to use your phone, you enter the old password, right? Because your brains like to operate at these really low energy states and anticipate what comes next. If that hasn't happened to you, there is something you can try, which is when you go home, change the location of the soap or shampoo in your shower, and I bet when you come back the next day, you actually reach for the old location. Again, our brains like to operate in these low energy states. And so the challenge is, how do we break free from this? Because I feel like that's really one of the keys to unlocking new ideas. And in my laboratory, we have been trying to harness a number of tools to break free from this repetitive thought process. And one of those tools is bioinspiration. And it's this idea that every living thing, every plant, every animal, anything that is alive around us today is here because it has solved an incredible number of challenges, right? Otherwise, it just becomes extinct. And so in essence, we're actually surrounded by solutions which I see as ideas for solving problems. Tens and hundreds of millions of years of research and development happening all around us. And we're only limited by our ability to go out into nature and make observations, and by the tools that we have available to uncover the mechanisms that nature has evolved to solve these problems. And what I find really remarkable is that tools constantly get better and better. We're constantly, research, we have new tools coming online all the time and higher resolution. And so we're constantly going to be uncovering new solutions in nature, new mechanisms. And so it's almost like this endless encyclopedia of ideas where new chapters are constantly being written. And so what I want to do today is share with you an example in the lab where we were working on a translational project. We literally hit a wall. We didn't know what to do. And we said, well, maybe the solution is already out there in nature. Maybe we can find ideas of creatures that have evolved to solve similar problems, and we can bring those ideas back into the lab and apply it to our problems. There we go. So meet Dr. Pedro Del Nido. He's the chief of cardiac surgery at Boston Children's Hospital. And he contacted me one late summer evening, and he said, Jeff, I'm trying to treat kids who have septal defects. These are holes in between the chambers of the heart. He said, sometimes I go to suture those holes, and the tissue is so fragile it just tears. And he says, that's not acceptable. He said, there's devices that work in adults, and they work fairly well. They're not perfect, but they work fairly well. But they're permanent. They're non-degradable. And you can't simply downsize those devices and put them into a child's heart, which is going to be growing over time, because it's unacceptable to have to come back every couple years and do these revision procedures. He said, is there any way that we could develop a patch that you could put up against the hole right here? It would immediately seal the hole, and the patch would then enable cells to migrate on top of it and form new tissue. So now you have the patient's own tissue on top of this patch. The patch would dissolve away, degrade, and the patient would be left with their own tissue sealing the hole, which would then naturally grow with the patient. So we were really excited to work with him, but we knew that this was going to be one of the most challenging environments that we would be developing a tissue adhesive, probably inside the entire body. because you have on average 60 beats per second, so that's a lot of expansion contraction cycles that we have to consider. There's a lot of shear force that's happening from the blood flow. It's extremely wet. There's enzymes. There's lots of cells that are present. So this is going to be a really tough challenge, tough problem to solve. So what we did is, in majority of translational projects that we start, we like to put together design criteria for a solution. So this is a list of properties of what we would see an ideal solution would have. And to me, this is, first of all, highly iterative. In many ways, you never fully nail it because you're constantly learning new things. But it's hard to do because it's easy to come up with a list of 20, 30 things, but then how do you narrow it down to 5 or 6, and then narrow it down to 1 or 2 that's going to drive a completely different solution than anyone has ever tried before? And so we came up with this short list here. We said, we're going to be placing this inside a heart. We're not going to stop the heart to get this patch to attach. So it would have to work in the presence of blood. It couldn't be fouled. A lot of materials that have been developed that are tissue adhesives, they actually react when they contact water. Or if they contact proteins, they get fouled. So the adhesive won't work when you try to push it up against the tissue. So we have to figure out a way around that. The material is going to have to fully degrade over time. It has to be elastic so it can expand and contract with the heart. Otherwise, it will delaminate and come off. The material has to be biocompatible, so cells have to be able to grow and form new tissue. And then talking with Dr. Del Nido and a number of other clinicians, they said, there's some materials that cure within 1 minute or 10 minutes in the clinic, but we don't like those because we don't want to be at the mercy of the technology. We want to be in control. If we're ready to seal this at 7 minutes and 32 seconds, we want to be able to do that. So we put on-demand adhesion as one of the criteria. And so this is a very practical consideration, something that I think we often forget in academia, are really the practical considerations of how is a surgeon going to be handling the materials that we're developing. And then also, because we're going to be curing this, and we thought using light activation would be useful, we've developed a lot of light activatable materials, we'd need some sort of a precursor form. So we have a patch and maybe a glue that is in a precursor form. We push it onto the heart, and we need the clinician to be able to move it around until they get it into the right position. And it can't wash out during that repositioning stage. So we said resist washout's another criteria. So we've been developing all kinds of materials in my laboratory, and some could fulfill some of these criteria, except for 2 of them. One is resistant to blood and resist washout. And so we really didn't know a way around this, and I think the field was really at a hold. We really haven't seen any major innovation in the clinic outside of sutures and staples, which have been around for decades and have tons of limitations. And so what we did is we stepped back and we said, how would nature solve this problem? Can we find examples of creatures out in nature that exist within wet, dynamic environments, similar to the environment inside the beating heart? And we found a number of creatures. We found sandcastle worms that exist in the sea, and you have the constant surf, and they remain attached to rocks. And then we have slugs and snails, which often you'll see in the rain are stuck on a leaf, and they're not falling off. They're able to stick under those wet, dynamic environments. And so we took a look at these, and we said, well, what do these creatures have in common? And we realized there were actually a couple key things. One is that they had viscous secretion. So anytime you see a slug or a snail, there's always this kind of slimy stuff coming out from the back. Well, that actually is part of an adhesive mechanism. Anything that's viscous has adhesive properties. So like honey on a plate, for example, doesn't easily wash away. And then when we took a look at these viscous secretions, we also noticed that they contain hydrophobic agents. And things that are hydrophobic can repel water. And so that gave us an idea. And we started thinking, well, what if we developed a tissue glue that was entirely hydrophobic? We would apply this to the patch, put it inside the beating heart, push it up against the heart, and it would repel the blood away from the surface of the heart. This would allow us to perform this procedure without stopping the heart. And then we said, well, if not only is it hydrophobic, if it's viscous, it's going to stay there long enough for the clinician to position it, and it would resist washout until the final cure. And so we spent probably 2.5 years developing this. But one of the things that we realized along the way is that just looking at slugs and snails was not going to be enough, and sandcastle worms. We needed to make this material super adhesive. And for that, we actually turned to ivy. And I don't know about Florida, but in Boston, there's a lot of buildings that look like this. I don't know about you, but I've actually gone up to those buildings and tried to pull the ivy away. And it's remarkable the amount of force that's required. It's really, really significant. So next time you're in New England or somewhere where there's ivy, give it a try, and I think you'll be impressed. So lucky for us, the mechanism through which ivy attaches to buildings was recently elucidated. And it turns out they have these root hairs, and these are like heat-seeking missiles. They go up and down the building, and they look for crevices. And when they find a crevice, they insert the root hair into it, they secrete a glue, and then they shrivel up and they mechanically interlock. It's remarkable. So that gave us an idea. We said, well, what if instead of just using reactive chemistry, which is what a lot of others were doing, and we had tried that previously with some of our tissue adhesive projects, what if we could develop a glue that would infiltrate into the tissue, just like ivy goes into a building, and then when we cure it, it will actually lock into place, kind of like a tissue like Velcro. And so with that in mind, we advanced. And literally took us 2 or 3 years. Many challenges that we faced. We had a highly multidisciplinary team, probably about 15 people on the team, biomaterials experts. We had a number of surgeons. We even had someone to help us deliver light inside a beating heart, a fiber optics expert. And so what I want to do is kind of fast forward to the most challenging experiments that we performed, and this is one of them. So before showing we could put a patch inside a beating heart, we wanted to start with the outside of the heart, the myocardium. And what I'm about to show you now is from an animal experiment. So if anybody is, gets queasy at the sight of blood or animal experiments, you may want to look away. So here, what we're doing is we're making a hole inside the myocardium here, the outside of the heart. This is a large hole, and this is a rat. And we have this suture here, which is just going to be a temporary suture. We're going to remove it at the end, and we're going to attempt to seal this hole with a patch and our glue. No staples, no sutures. And you'll see that this is a very high-pressure environment. We're not going to wipe away the blood. There's the patch, which is also biodegradable, and it's transparent. There's glue on the underlying layer. We push this up against the heart. We shine the light for 5 seconds. And while the patch attached, this is one of the early animals we treated, we had something catastrophic happen, which is the patch kind of slipped away from us because we were nervous, and there was a leak. Completely unprepared for what to do, we just scrounged up as much glue as we could find, and we applied it directly to the leak. Yep. And you'll see that if you look carefully, it's less red where we've applied the glue. It's actually repelling the blood away because it's hydrophobic. And because it's viscous, it stays in place long enough for another pulse of light, 5 seconds. Remove the suture, and we have a perfect seal. Okay. And we took these animals out 6 months, and they did fine. And so now what we wanted to do is move to an even more challenging experiment, which was go from a small animal, a rodent, to a large animal, a pig, and see if we could get this to attach inside a beating heart directly to the septum, where these kids tend to have the holes in between the chambers of the heart. And so for this, Dr. Del Nido had developed a device for delivering all kinds of different devices into the heart. And what we did is we attached the patch to the end of the device, made a small incision in the myocardium. This is a proof of concept experiment. We applied this to the septum. We shined the light for 20 seconds. We removed, we sutured the myocardium. And this is what it looked like from the echocardiogram immediately after the procedure. So it's 2 separate pigs, and you can see the patch is attached. The heart rate here is 82 beats per minute. We came back 4 hours later and added epinephrine to increase the heart rate to cover the full physiological range. And you see here that heart rate's 165 and the patches remain attached. If it wasn't attached, you wouldn't be able to see it, it'd be moving around so much. So then we came back after 24 hours and the patch was still there. You can see a suture here, which was part of the deployment mechanism. And you can see here that we have after the 24-hour time point, we have a thin capsule that's starting to form here. So for us, this was really a major step forward because it demonstrated that we could get this patch to attach inside a beating heart during a procedure in a large animal that mimics many aspects of the human condition. And so we were pretty excited about this. And you'll notice, though, that we had to make a hole to seal a hole. So that's not, this is really just a proof of concept experiment. And we were constantly, we have our eye on the clinical application, not just publishing papers. And so we stepped back and we said, you know what, we really need to solve this problem. And so we teamed up with Connor Walsh at the Wyss Institute in Boston and continued to collaborate with Dr. Del Nido to develop a way to deliver the patch through a blood vessel into the heart so that we wouldn't have to make this second hole. And so I'm going to show you an example of how this device works.
Researchers from 4 top institutions in Boston have designed a specialized catheter that can repair holes in the heart without invasive open heart surgery. First, a clinician inserts the catheter, which is equipped with a UV light and biodegradable adhesive patch, through a vein and guides it to the hole in the patient's heart. Next, the adhesive patch and 2 balloons, one on each side of the hole, are released from the catheter. The balloons apply pressure from both sides of the heart wall, securing the patch in place. Then, the clinician turns on the catheter's UV light, which reflects off the balloon's interior and activates the glue on the side of the patch facing the heart wall. Finally, the clinician deflates the balloons and removes the catheter, leaving behind the biodegradable patch. In time, tissue will grow over the patch, and the patch will dissolve, leaving no foreign material in the body.
So this was really a way to solve the problem of creating a hole to seal a hole. And this is now a developed and functioning device. We recently published a paper on this demonstrating that it could work in a pig model. And so we're thinking of translation. And so as we're considering this application of sealing septal defects, we recognize that the bar is actually very high for treating pediatric patients. And so we continue to advance this in the laboratory, but because this material is so promising, we showed that it could seal the carotid artery of a pig, the aorta could stick to intestine tissue. We formed a company called Gecko Biomedical in 2013. And the company has actually taken the technology from my lab and created a stable, a scaled product. And here's what it looks like. So Bloomberg came by the lab not too long ago and created this video. And you can see we're applying the glue to a vascular graft that it attaches very well to it. You see the precursor is a viscous kind of honey-like material. And this is Maria, who was a PhD student in my laboratory, who co-developed this material with Nora Lang, who was a cardiac fellow in Dr. Del Nido's laboratory. So the two of them together were really the leaders on this project. And now Maria is head of research at Gecko Biomedical, which is based in Paris. And the company now has been able to advance this technology that we developed in the lab and is currently testing this glue for vascular reconstruction in human patients in Europe. So I was talking a little bit about this, this glue when I was at a conference in Boston that was at the New England Aquarium. And I'll never forget that conference because it was right where anyone who's been there knows that there's a penguin exhibit. And it was literally like right in front of the penguins, which were kind of behind. And they were so noisy, I could barely even yell loud enough for people to hear what I was saying. But this guy was in the audience, Dr. Rosenblad, and he heard a few things that I was talking about. He's head of dentistry at Angel Memorial, which is really a top-tier vet clinic in the New England area, and he said, I think your glue could be very useful for treating not just humans, but actually animals, and I'd love to talk more with you about this. So about 4 or 5 months ago, he contacted me again, and he said, I have a dog, a patient that's a bulldog. It's 10 years old, and it has an oral nasal fistula, a hole from the oral cavity all the way to the nasal cavity. He said, it's so big I could put my finger right through. And he said, the dog's in miserable shape. It's like on its last legs. It's getting infected. He said, is there any way we could apply your tissue glue to fill the hole and help this dog? And so the concept was that we would need to create multiple layers of this glue. We were going to cure the glue right into the hole after debriding it. And then we were going to pull a tissue flap. We'd cure the glue and then pull this tissue flap over top and see if we could get that to work. And he had performed 3 surgeries on this dog, and all had failed within 2 or 3 days. He had just tried to place a flap over the hole, but because there's so much tension, especially in the mouth of a bulldog, it was just tearing open after each procedure. So you can imagine the owner and the dog and the vet have been, by this point, very, emotionally distressed about, just really trying hard to do whatever they could to save this dog. And so we had never tried this glue could work on in the oral cavity. And so Yuhan Lee in my lab, who's leading this project, next day we I talked to Dr. Rosenblatt, and the next day he actually brought this cow head in the lab that he got from a local butcher. And here you can see that he's actually applied the glue to the oral cavity, and you can see it's actually sticking really well. In fact, he's taken like a metal spatula and is trying to remove it. So we knew that this glue now could attach to oral mucosa, and so we went ahead with the procedure at Angel Memorial. And so here's what the fistula looked like. It was actually created from when Dr. Rosenblatt pulled a tooth and it didn't heal. And you can see it's infected here, lots of hair and all kinds of junk that was in that space. So they cleaned that out. We cured the glue in this spot, and then Dr. Rosenblatt created a tissue flap and pulled it over top of the hole and then sutured it. And about 2 weeks after the procedure, the first 3 procedures that were performed within 2 to 3 days had failed. But after 2 weeks, the dog came back, and you can see that the flap is still there. And literally, Dr. Rosenblatt and the owner were almost like crying. They were so happy because they had been through such difficult time. We were caught a little bit off guard because we just weren't in the same mindset as they were. And the dog, actually, this is what the dog looked like before. So you can see, not very happy. And then this is the dog afterwards. You can see a remarkable difference. And this dog, actually, the name of the dog is Poppy, named after Big Poppy. So anybody who follows baseball knows of Big Poppy, who's the star, just retired this year from the Boston Red Sox. And so, I was calling the dog Little Poppy. And I think there's actually some resemblance to Big Poppy. And so here's the team here, and it's been now over 3 months, and Poppy's doing really well. So here's the team, Dr. Rosenblatt. There's Yuhan, who's leading the project, and some interns in the lab. Here's a technician from Angel Memorial, and here's the owner of the dog, Mr. Shanahan. And this actually, the press got word of this story, and they came to interview Little Poppy. And so you can see he's kind of having his glory day here at Angel Memorial. So to me, this was actually a really exciting moment in my career, and also the people in the lab who are part of this project, because we had always thought that the impact that we would secure at some point in the distant future would be on humans, and we never realized that there was this amazing opportunity in our backyard, literally, to help animals as well. And so now this has become one of the missions of the lab, and I've actually gone over to Angel Memorial and talked with a number of vets over there. I gave a lecture in their lecture series, and I'm trying to figure out ways that we can move some of the other technologies in the lab to help animals in a shorter period of time. So I just want to share with you one other tool that we've used. I talked a little bit about bioinspiration, but we also use another tool in the lab that I like to refer to as radical simplicity. And again, it's really this notion, the paradigm I've tried to establish is to not just publish papers, but develop technologies that can help patients in the shortest period of time. And I can tell you so many stories that have happened where we just developed really complicated approaches that never made it out of the lab. And I think it's this notion that in academia, there's almost this pressure where the more complicated the technology you develop, the greater the chance you have of publishing in a good journal. But the challenge then is you have a lower chance of actually helping anybody, because it's the simple technologies that can get, that can advance the fastest to help patients. And so, so what we've been really doing in the lab is trying to find ways to keep the technologies as simple as possible at every single stage. So let me give you an example that is focused in the area of stem cells. So one of the big challenges in cell-based therapy, and in particular for stem cell-based therapy, is that we can grow stem cells, almost any type of stem cell now, in a petri dish, and we can control almost everything we want. We can pattern the media, we can pattern the substrate, we can get the cell to do anything we want. But the challenge is that when you go to transplant those cells in the body, you lose all of that control. They're entirely at the mercy of the biological milieu, and if the cells end up in different sites in the body, they're going to behave completely differently. And so we've been interested in developing technologies that can control cells following transplantation using very simple approaches. And I'm happy to say that Professor James Ankrum, whose picture here is right in the middle, is in the audience today, previous PhD student from my lab who's now faculty in Iowa. And so what we did is we looked at this and we said, there's all kinds of complicated things that we could do. And it was really the first thing that we thought of was, how could we solve this problem? And it was all these complex ideas came to us, but we really wanted to keep it simple. And it took us a while, but we eventually figured out a way to control cells following transplantation using perhaps one of the simplest technologies you possibly could imagine. So there's all kinds of degradable materials that already exist in the clinic right now, like degradable sutures and pins and plates, screws for orthopedics. And we actually end up using some of those materials. And the concept here was, how could we deliver devices that would be internalized into the cell and deliver molecules to the cell after it was transplanted. So that was really the concept that we wanted to advance. And so what we did is we discovered that particles that were around 1 micron in size were readily internalized by certain types of stem cells. We could put small molecules into those particles, and those particles would be internalized into the cell, and we found a certain size range where those particles would stay in cells for weeks and weeks. And what would happen is we could control the degradation of that material. These are FDA-regulated materials that we're using, already scaled to very, very large-scale manufacturing. And we could put in any molecule we want and deliver that. As the material was degrading, it would release the molecule to the cell, and we could activate pathways in that cell to control it after transplantation. And so here what you can see is in green, we have the particles, and the red is the cell membrane. So you can see these particles are inside the cell, and we were able to show we could keep them there for weeks and weeks. And not only were we interested in controlling the cells, we thought, well, if we could develop this type of technology, maybe we could control the environment around the cells as well, which is extremely important to consider. It's not just about the cells doing their function, it's the cells interacting with the environment around them, and we wanted to control that as well. And what we found is that by putting these molecules into the particles that were inside the cells, as these particles degrade, because they're made out of degradable suture material, the molecules were able to activate the cells, but also cross the cell membrane and interact with neighboring and distant cells. And so we were able to show that we could control all kinds of different properties of cells and neighboring cells, and we've published a number of papers on this technology. We've shown that we can boost the immune modulation of mesenchymal stem cells as one example. We can improve the imaging of the cells following transplantation. We can control the differentiation, and the list goes on and on. Again, this is a very simple technology. Microparticles, nanoparticles already exist in industry, in the clinic. The materials we're using are already FDA regulated, and the molecules we're using are also already FDA regulated. And so really it's just this combination of technologies that already exist has created a very powerful solution so that now we can control cells following transplantation, and we're exploring this for a wide range of therapeutic applications. In addition to this, we've also been interested in applying this concept of radical simplicity to drug delivery. And so, most people are familiar with taking a pill. And the challenge is that, this is a standard mode of therapy is taking a pill, but then you have to bypass the stomach. You have stomach acid there. You have to get it into the gut. It has to be absorbed. Sometimes it goes, through the liver and it gets degraded. And so there's all sorts of barriers that the drug has to go through. So you end up getting a very small amount that ends up in the circulation, and then it goes throughout your body. But often you only want that drug in a specific part of your body, localized. And so there's huge unmet need for all kinds of therapeutic applications where you'd want better control of drugs locally. And we wanted to know whether we could apply the concept of radical simplicity to drug delivery. And this would have implications for arthritis, ulcerative colitis, all kinds of different disease states. And so, again, we initially thought of these complex approaches. We stepped back, we stopped ourselves, and we went to the generally recognized as safe list by FDA. And this is a list of agents that exist in foods, they exist in cosmetics, and they've been deemed safe by the FDA if you apply them topically or if you use them in certain concentrations. And the nice thing about this list is these agents exist in ton quantities, GMP grade. And so what we did is we scanned this list and we identified small molecules that had hydrophobic and hydrophilic domains, so water-hating, water-loving, and an enzyme cleavable bond. And I'll explain why in a moment. We found these on that list. And what we were able to do is take those molecules and put it into water, and by coaxing the system just very simply with temperature and with some solvents, we were able to get that single molecule to stack over and over and over again and form a gel-like material that looks like this under electron microscopy. Now, what you're looking at here is a single molecule that's stacked over and over and over again. There's no excipients present here. This is not a polymer. These are non-covalent interactions. It's a single molecule that's just stacked and interacting with itself. And during the assembly process, we can incorporate all kinds of drugs. We figured out how to do hydrophobic, hydrophilic, and now even biologics like peptides and proteins. And then in the body, in response to inflammation, so patients who have arthritis, for example, there's many cases where you have a flare and then a period of remission. And so if you have a drug delivery system, you only want it to be releasing drug during the flare and not during remission, because then you're just depleting your stores. So we created this system so it would be enzyme responsive or inflammation responsive. So in the presence of inflammation, you have all these degradative enzymes, and so that look like this. And what happens is it breaks down these molecules, this enzyme cleavable bond, and releases the payload. And we've shown that this has potential to treat all kinds of different diseases, such as ulcerative colitis, arthritis. We've been applying this to limb transplantation. The list goes on and on, a number of types of cancer. So we've tested this in 6 different, now actually I think it's 7 or 8 animal models. A number are large animal models, including pigs. And really, the advantage of this technology is that it's very simple. We're not synthesizing materials. We're just buying them from the generally recognized as safe list. And we recently started this company called Alivio Therapeutics, which I co-founded with Bob Langer at MIT, as well as PureTech Health, which is a VC firm in Boston. And we've set up our lab, and we're moving full speed ahead to the clinic. So if all goes well, this could enter human testing within the next 2 or 3 years. And then finally, I just want to end with one last example of bioinspiration. I want to come back from radical simplicity to bioinspiration, because we were really interested in asking the question, could we apply bioinspiration to tissue regeneration? And when you think of tissue regeneration, you really think of, it's really a holy grail. But if you step back and kind of look at the animal kingdom and nature, you realize that there are many, many examples of tissue regeneration occurring in adult creatures across the planet. So we know that sharks are able to regenerate their teeth throughout life. We know that lizards, if you chop off the tail, will actually regrow the tail, and the list goes on and on. And so the question, though, is that why don't humans have these abilities? So if we see these abilities all over the animal kingdom, why don't we have those? And the answer is, and I think, as we think about this a little bit more, perhaps when you look at nature, we have the building blocks out there. We really just have to decode the blueprint, right? And when we look at our bodies and we study biology, we realize that we actually do have many instances where tissue regeneration is already occurring in everybody in this room, in fact. So the intestine, the lining of the intestine, is one of the most regenerative tissues that has ever been discovered. It completely regenerates every 4 or 5 days. And so we were really intrigued by that, and we wanted to see if we could identify some of the cues that were responsible for this regeneration. And so what we did is we turned to the intestinal stem cell. So there's a stem cell that exists at the base of crypts in the intestine, which are labeled green here, and the red that's the intestine. You see the villi and the crypts. And these stem cells actually are the powerhouse behind creating the epithelium, the lining of your intestine, the powerhouse of creating this new epithelium every 4 or 5 days. And so we stepped back and we really looked at the signaling that goes on within these cells and the neighboring environment, because what we wanted to do was control these cells. If we could control these cells, we might be able to activate tissue regeneration in other places in the body. And so what we did is we looked at the pathways, we found small molecules that could activate these stem cells, and we were able to take a system like this and produce nearly pure populations of intestinal stem cells simply with small molecules. And so we published this work a few years ago, and we've been looking at other systems to apply this to. And recently, about maybe 3 or 4 years ago, we started looking at the inner ear. And what's amazing is that these intestinal stem cells are marked by a ligand called LGR5. And that marker was discovered within the inner ear. And we thought, if we could apply the knowledge that we've gained from the intestine to the inner ear, maybe there's an opportunity to restore hearing. So each person, everybody here was born with 15,000 hair cells per inner ear. And you essentially die with the same cells. They never regenerate after you're born. And due to noise damage and various types of drugs, those hair cells are being killed all the time. So as you go on in life, the number of hair cells you have decreases, and so does your hearing. And so what we did is we were interested to say, could we now take this understanding of the basic cues in the intestine, this highly regenerative organ, and apply it to the inner ear? And what we found is that we could take our molecules, apply it to the inner ear. We were able to proliferate the stem cells. There's a supporting cell, a progenitor type cell within the inner ear that's the progenitor of hair cells. We were able to find molecules that could amplify those progenitor cells and produce tons and tons of new hair cells. And that's what you're looking at here. You can see these pristine hair bundles, which are responsible for interacting with sound and then converting it through connections with neurons into signals your brain can interpret as sound. And so we've now found that we can promote the regeneration of hearing in animals, and we've formed a company based on this technology called Frequency Therapeutics. This is really a platform. The inner ear, so to restore hearing, is the first application. And we're in the process of moving this very rapidly towards the clinic. But this is a platform, and we can apply this to many other sites in the body. And this company, Frequency Therapeutics, is just in the process now of coming out of stealth mode. We formed it a couple years ago and have been very busy moving this technology and building the team to advance this to the clinic. So in summary, today I talked about some of the tools that we've been harnessing in the laboratory, namely bioinspiration and radical simplicity. For bioinspiration, this is not biomimicry. We're not just trying to mimic creatures that we see as solutions, every single detail. We're actually just taking a basic idea from nature and then improving on it for our own purposes. And then radical simplicity is really stopping ourselves from trying to do the traditional academic process and develop these complicated technologies, and really step back and say, how can we keep this as simple as possible at every single step? And really think ahead the full translational spectrum in terms of scalability and manufacturing of the technology. So at this time, I want to acknowledge the wonderful people in my laboratory who have created the data that I've shown you today, and all really wonderful people, all my collaborators, of which there are many. And I'd like to thank you for your attention and my various funding sources, and I'm happy to answer any questions you may have. All right, thank you. Are there questions? I think it's on now. I can hear you, yep.
My question is, with regard to the hydrophobic, hydrophilic, or amphiphilic grasp substance that you mentioned, how does it interact with cells, or maybe how do cells respond to being in that matrix that you showed? And what is the substance as well?
Yeah, so the question is, how do cells interact with these gel-like materials that we've been developing from generally recognized as safe agents? And what exactly are those materials? So I'll answer the second question first, and then I'll come back to the first one. So we've found molecules like ascorbyl palmitate, which is long-acting vitamin C. It has a hydrophobic and a hydrophilic domain and an enzyme cleavable bond. And so you can go to the drugstore right now and buy that. And all we've done is change the format. So instead of that molecule existing in like a free format once it gets into solution, we found a way to create a gel out of it. And there's many examples like that. Another example is we've taken an emulsifier that's in ice cream, and we've been able to create a gel from that. So there's many different molecules that we've shown this works for. We've also been looking at, so these are on the generally recognized as safe list, which typically means that they're safe in certain concentrations when you put them on the skin or if you eat them, but not necessarily if you inject them into the body. And so we've done a number of studies where we've looked at the biocompatibility of these materials, and we found for the most part that we have very good biocompatibility. So we've done head-to-head comparison with degradable sutures, so which is polylactide coglycolide. And in most cases, we have much less inflammation than the existing degradable materials that exist in the clinic. Yeah, so we haven't really advanced many projects yet where we've mixed cells with these gels. We've typically been looking at this more as a drug delivery platform, an inflammation responsive system. So for treatment of ulcerative colitis, as well as arthritis and limb transplantation. But this is something that we're actively looking at, is whether we can. So for example, we've taken tacrolimus, which is an immunomodulating agent for limb transplantation, and we've put it into the gel and shown that we can, with a single injection in a transplanted limb, we can keep it alive from 100 to 150 days. We think that this system could be very interesting for transplanting allogeneic cells, so for example, beta cells, which may get quickly rejected. So if we put them into a gel that has an immunomodulatory agent in it and can be released for long periods of time, that could be an interesting platform, and that's something that we're looking at now.
Yes, I was just wondering about the ulcerative colitis. You were talking about how the cells regenerate in the intestines, but then there's a chronic inflammation.
Yeah.
With it. So if you do actually add stem cells in there, what will they do in ulcerative colitis, and when will you have a treatment for that?
Yeah, so ulcerative colitis is really a tricky disease because, well, there's a lot known, there still is a lot unknown in terms of the systemic effect of a systemic inflammatory effect versus local effect. There has been studies where people have taken these LGR5 stem cells that exist within organoid structures, which is just like a ball of cells, and a small fraction are the stem cells, and they've done infusion, enema infusion in animal models of ulcerative colitis. They're able to attach and engraft and accelerate the healing process, but the animal models of ulcerative colitis are not very good predictors of what's going to work in humans. It really only mimics, one aspect of the disease and not the background inflammatory response. And so I think right now, at least my thinking is that for stem cell therapy to work in the context of inflammatory bowel disease, you probably will need to have an immunomodulatory agent as well as the stem cell, to complement the stem cell therapy. What we've done, what we focused on with our drug delivery system is almost every ulcerative colitis patient will require an enema at some point, enema treatment at some point in their therapy. And that's problematic for a number of reasons. One, they have to hold it in, which is really challenging to do. Second is that you have a lot of systemic absorption of drug, so there can be toxicity associated with that. And third is the dosing is every day, so compliance can be very low. So we showed if we formulate these gels with a steroid, just as a first example, that we could get and break the gel into particles in a suspension, an enema suspension. We showed in several animal models that this could attach rapidly within seconds to minutes to the ulcers, so the patient wouldn't have to retain the enema. We showed that we could reduce the systemic exposure by 5 to 10 times, and we showed that we could dose every other day instead of every day with our system, because the gel attaches to the ulcers and stays there and continuously releases the drug right at the site of the ulcer. So we are advancing that. So we're looking at that actually from two separate angles. One is drug delivery, and one is from a stem cell-based therapy. Other questions? No? All right, thank you so much.